Hello, welcome to the Masterclass on how to extract high-quality DNA and RNA. My name is Vania Costa, and I'm a field application scientist at Oxford Nanopore Technologies. In this Masterclass series, we are walking you through the whole Oxford Nanopore workflow from sample to answer to ensure that you have support all the way through. Now, this Masterclass is focused on how to extract high-quality DNA and RNA. In this masterclass, we'll cover topics including how to choose the best extraction method, how to ensure you have high-quality nucleic acids, how to optimize your extraction for your experimental goals, how to QC your sample, and finally, how to handle and preserve the nucleic acids. To kick things off, let's discuss what you'll have to consider before you get started. Usually, when people think about an experiment, they tend to think about their sample, then they do an extraction, then the library preparation, then they generate the data, and finally, they see if they can answer that scientific question, when actually it should really be the other way around. So you start off by considering what type of data you need to answer your question and work backwards because what you need from your data will impact the library prep method you will use and how you extract your DNA and RNA. And often the sample you use to start with will have an impact as well. So before you get started with your experiment, I really recommend that you consider depth of coverage you will need and the read lengths you wish to produce in sequencing, because different applications will have different requirements. Then you can choose a library preparation protocol and an extraction protocol that are suitable to answer your questions. So today, we'll be going through the first steps of your nanopore sequencing workflow in detail, and I'll show you where to find documentation for our verified extraction methods online. One thing I wanted to share, though, is that we also offer application workflows, and these are protocols that take you from sample to answer. So you'll start with the extraction step, but then the protocol will take you all the way through library preparation, sequencing, and even data analysis, so you will be supported all the way through your experiments. There's a variety of things you should consider before you begin your experiment. First, there's the starting amount. You'll need to choose an extraction method that will yield enough DNA or RNA for the library prep method you're planning to use. You'll also need to ensure you store the samples and the extracts correctly to avoid any type of degradation. Then, when it comes to the extraction itself, the sample type will, of course, have an impact because different methods are optimized for different samples. And then there are samples that are they just naturally contain some contaminants or anything that can be carried over. So, for instance, blood has a lot of globulin. So, if you are extracting RNA from blood, then you will want to deplete the hemoglobin so that it won't dominate the reads you are generating. Some samples might have an extra layer of complexity. So for instance, mixed microbial samples, they might require specific methods. And then a key part of sample prep is making sure that your DNA or RNA looks as you need it to look to answer your scientific questions. And using sample manipulation like size selection or fragmentation, you can enrich for certain fragment lengths or shear your DNA to make fragments shorter. This is something we're going to go into a bit more detail in a few minutes. And finally, if you want to sequence the samples in multiplex, you'll need to consider how many samples you can pool and sequence in a single run. To answer this, again, you'll need information like the read length and the depth of coverage you will need from your data. And all of these considerations will help ensure that you get the data you need. With Oxford Nanopore sequencing, read lengths are unrestricted. So you can sequence anything from short fragments all the way up to ultra-long fragments in one single read. And when we talk about fragment length, we tend to divide them in three categories. For short fragments, we're talking about fragments down to 50 base pairs. And some samples that you wish to sequence, they will just naturally be short and degraded. So let's think about cell-free DNA as an example. It's just naturally very short, and that's absolutely fine. If you put these short fragments on your flow cell, you will get the short reads out. It's still going to sequence them. And then there are long fragments, so things up to 100 kb. An important consideration is that there is a trade-off between maximizing output and maximizing read length in sequencing. So, for example, if you're looking at methylation, you probably want to maximize sequencing output, but read length is not as that important. So perhaps you want to fragment your sample to make sure you have a shorter read length than 50, but you generate more data. However, if you're looking at, for instance, sequencing structural variants end-to-end and performing phasing, then you probably want longer reads, let's say about 30 kb. And finally, Oxford nanopore sequencing can sequence ultra-long fragments, so fragments over 100 kb, all the way up to megabase in length. For applications such as assembling complex genomes, generating telomere-to-telomere assemblies, and resolving really complex structural rearrangements, ultra-long reads are key. Now we are going to talk a little bit about sample handling and storage. This is really important because the number one rule of extraction samples is that you cannot sequence what you do not have. So what you put into your nanopore sequencer is what you're going to get out. So if you start with a fragmented sample, you're not going to get long reads because you don't have those long fragments to start with, so you can't sequence them. Everything from sample storage and handling to extraction and manipulation will have an impact on the data you are going to generate. So it's important to start your experiment with the right extraction and sample processing. Here is an example of the importance of optimal sample preservation. So on the left, you'll see the read length N50s for DNA that was stored in 3 different ways before I extracted the DNA. It was sequenced straight away, so the control sample, and sequenced after storage at room temperature or at 4 degrees. And as you can see, there is a reduction in the read length N50 for the sample kept at room temperature, showing that the sample degraded. Room temperature is not good to store samples. And then in the example on the right, there are human cells that were preserved at -80. So they were collected and preserved as freeze-dried in glycerol and then in a mix of FBS and DMSO. And what we saw is that the samples stored in the mix of FBS and DMSO produced a higher read length N50 than the others. So in this case, it's not just about the storage temperature, but also about adding a preservative solution that will ensure the samples remain stable. So as a general rule, for both DNA and RNA, you should try to keep the samples at -80 whenever possible. This is the safest option. And if there are extra solutions you can add to help preservation, then that's great too. There are a lot of commercial solutions available for any sample type you can think of. And here you can see just some of those examples. So you should think about the sample you have and what's the best way to preserve it. Once you've extracted your samples, then you need to preserve the DNA or the RNA in the best way possible because you also want to keep that intact. And in this example, we extracted bacterial DNA and then stored it at 4 degrees and at -20. And what we saw is that for short-term storage, 4 degrees was fine. But for long-term storage, we saw a drop in the read length N50. So long-term storage, -20 is the best option. It's also important to note that we've seen this with DNA eluted in T, so EDTA was present. If you repeat the same experiment without any EDT present, then the drop on the fragment length could be even faster than what we observed here. When handling DNA samples, if you wish to preserve the really long fragments, you should use whiteboard tips and mix by flicking the tube. You should not vortex the tube. We also don't recommend pipette mixing, but if you really have to do it, then do it slowly and again with whiteboard tips. When you freeze a sample, it is possible that you will subject it to freeze-thawing cycles. And as you can see on the right, we've tested the effects of freeze-thawing as well. So up to 10 freeze-thawing cycles, we didn't really see a big change on the fragment length, but after that, the fragments start getting shorter. which means that the sample is degrading with free-thawing cycles. So ideally, and especially if you have a lot of DNA, you should try to keep it in aliquots just to kind of avoid multiple freeze-thawing cycles for each of those tubes. Um, and just remember to also keep your DNA samples, with a pH within the reasonable level, avoid high temperatures, and of course, avoid introducing DNases because they will destroy your DNA sample. And then for RNA, we've also assessed its stability at room temperature 4 degrees and -20 in TE, water, and RNA storage solution. What we've seen is that there isn't a big difference between TE and RNA storage solution, but there is a drop in RNA integrity number, or RIN, which we'll talk about in a little bit. And we've seen this drop for the samples stored in water. And just to note, in this instance, we did not sequence the samples. We just performed a QC, and it was obvious that there was degradation on the samples preserved in water, especially if kept at room temperature. For RNA, we recommend -80 for long-term storage, and again, using aliquots to avoid freeze-thawing cycles. It's also important to avoid introducing RNases because this will destroy your sample. And to do this, it's really important to ensure that samples, consumables, the lab benches, everything is RNase free. And then when thawing the samples, keep samples on ice to avoid harsh temperature variation. The high temperature should be avoided, and the pH should also be stable. Now let's talk a little bit more about how to choose the right extraction method. So for research samples like cells, blood, tissue, you tend to have a variety of kits that work with pretty much all the samples, not just mammalian, but the things like, for instance, flies or worms. And you can choose a kit that will allow you to get fragments within the range you want. So for instance, if you are interested in shorter fragments, my recommendation would be to use a spin column-based method like ChIAMP or DNeasy, which will result in fragments within the 10 to 20 kb range. So you don't need to process your sample any further. You can just use it as it is from the extraction. ChIAMP and DNeasy are very similar chemistries, but DNeasy is optimized for smaller input sizes. And if you use a method that will generate longer fragments, like for instance PureGene or QIAGEN genomic tip, you will probably want to combine it with light shearing just to make sure your sample it's easier to handle. But then if you want longer fragments, you can use those same kits, so the QIAGEN, PureGene, and the genomic kit, and combine them with size selection to enrich for longer fragments. Then there are specific kits like, for instance, the NEB Monarch, which we will use for the preparation of ultra-high molecular weight DNA. And this will give you like these beautiful ultra-long fragments. But if you're using something like a ligation-based library preparation approach, this will be a little bit too long to be efficient with that preparation. And this ultra-long prep I just mentioned will be covered on the next masterclass. So in this case, you might want to fragment the sample a little bit just to make it efficient, but as I said, we will go into the fragmentation in a few minutes. And then there are just a few considerations, like for instance, if you are working with nucleated red blood cells, you will only need a very, very small amount of sample because the red cells have nuclei, which means you're going to generate a lot of DNA from a very small amount. And you have to ensure that you use a low amount and that you dilute the sample to make sure you don't overload the kits that you are using for extraction. For samples that have tough cell walls, we recommend different approaches. So for plants, we recommend CTAB and beta-mercaptoethanol, which are chemicals that are a little bit harsh. So then we recommend doing a cleanup with a QIAGEN genomic tip. It's just an easy purification method to ensure that no contaminants are carried through the extraction. Then for fungal species, we recommend a PVP and SDS-based approach. and then just precipitation cleanup. For mushrooms, we recommend nuclei isolation. We've seen that it works really well, but it also involves some harsh chemicals, and we recommend a QIAGEN genomic tape purification after the nuclei isolation and extraction. Then for bacteria, the kits that you can use are pretty much the same as you would use with the samples we discussed on the previous slide. The difference is that you need to use a specific enzyme. So these kits tend to have the enzymes incorporated to make sure that you can blast the bacterial cell walls. For environmental samples, we recommend bead-beating based methods, especially if you don't know what's in your sample and you want to extract everything that is in there, because mechanical lysis will extract all that is present. Something enzymatic might not be able to burst the cell walls of every species. And then you can also combine this with host depletion to get rid of any unwanted host DNA. Then there are some specific sample types for which we recommend specific kits. For example, for FFP samples, cell-free DNA, plasmids, or blood spots on FTA cards, you would use a very similar protocol to what you would use for soft tissues, but with just some adaptations. Now, all of these protocols have been verified for use with Oxford nanopore sequencing and are available on the documentation section of our website. For RNA, the situation is a little bit different. So for DNA, kits often tend to work with a variety of samples. With RNA, the kits tend to be sample specific. So we recommend the QIAMP DNA FFP tissue kit for FFP samples. Then for blood, we recommend the collection in packaging tubes, and then the same kit for extraction. And in the case of blood, we would really recommend that you perform a globin depletion step before you proceed to library preparation. So this is the case I mentioned previously, where if you sequence it as it is, so without the globin depletion step, the majority of your reads will be globin. And then for cell lines, we recommend Trizol, which can also be used for other samples like worms or yeast. Then we also have some specific recommendations for plants, animal tissues, and mushrooms. Again, you can find all of the protocols on our documentation section in our website. For bacterial samples, we recommend different kits for gram-positive and gram-negative bacteria. It's important to note that our RNA kits, both the direct RNA kit and the PCR cDNA kit, they require poly A tails, and bacterial RNA doesn't naturally have a poly A tail. So once you extract the bacterial RNA, you need to perform a ribodepletion step and then a polyadenylation to make sure that your sample is ready for library preparation. Once you have extracted your sample, there are other steps you can take to make sure your DNA or RNA looks the way it needs to. So I previously mentioned manipulation of the sample, and that's basically fragmentation and size selection. Now, starting with fragmentation, there are several reasons why you would want to include this step. The first one is to optimize for sequencing output, because if you have shorter fragments, you're going to generate more data. So if you're aiming to maximize output rather than optimizing for longer reads, this would be a way to do it. Secondly, fragmentation is used to generate optimal fragment lengths for ligation-based library preparation. I mentioned the NAB Monarch kit, which produces ultra-high molecular weight DNA. And this is usually a little bit too long for a ligation-based library prep. So in this case, just fragmenting it a little bit will make sure that the ligation reaction is efficient. Then it's also useful just to fragment really viscous DNA to make it easier to handle and quantify, because it's almost impossible to accurately quantify a sample that is viscous. And then again, it also helps ensuring that the library preparation is efficient. And then it's good to keep in mind that our transposase-based library prep approaches include fragmentation, and there will be more information in the next masterclass of this series. The basic rapid library preparation method, it just takes about 10 minutes, so it's really, really fast. And this is the same chemistry that is used for the ultra-long DNA sequencing kit. And it's particularly useful if you're working with plasmids because the transposase linearizes the circular molecules. So you avoid the linearization step before the library preparation as it's already included on it. Now let's talk about DNA fragmentation methods. On the left, we use Megaruptor, which is a needle-based technique. And this is a good example of why fragmentation can be useful. So with this method, you can choose the speed at which you want the DNA to pass through, and the faster it goes through, the more it shears. So while the unsheared sample and the sample sheared at speed 3, so really slow, produce really long fragments, you can see here that the read length N50 is about 10 kb, while the samples sheared at the highest speeds to produce shorter fragments produce read length N50s up to 20 kb. And this is because the first two samples contain fragment lengths that were too long to go through this particular library prep method, which uses the ligation sequencing kit. And they will be too long to go through it in an efficient way. And this is why if your DNA is too long, you should shear it a little bit to make sure you get the most out of it. Another method we recommend for fragmentation is the Covaris G-tube. So in this case, you can just pass your DNA through a tube at a different rotation speed in a centrifuge, and this will produce different fragment lengths. This can be useful for where you don't have enough starting material. So of course, we always recommend that you start with enough material, that you put enough material in your library preparation. But sometimes we know that it might be a bit difficult because you might not have more sample to re-extract, and you just have the DNA you have, and you have to use that. So with oxo nanopore sequencing, what matters, it's basically the number of ends. So it's the molarity that will actually impact the efficiency of the attachment of sequencing adapters. So if you have really long DNA fragments, you're going to need more sample. Whilst if you have shorter fragments, you don't need this that much. And this is why if you have a low starting amount and you cannot re-extract anymore, there's nothing else you can do. You might consider fragmenting it because then you will be able to reach the necessary molarity. You can also enrich for long fragments, and to do so, you use size selection. So to deplete fragments up to 25 kb, we offer the short fragment eliminator kit. This is really easy to use. So you just mix the solution with the sample, and then you just spin it down, you wash it a couple of times and elute, and that's it. So it's a really fast protocol for enriching long and ultra long DNA fragments. There's also other options available for the depletion of different ranges of fragment lengths. One is private, which you can use to deplete fragments up to 1.5 or 2 kb. There is also the blue pippin, which is customizable, so you can select the fragment lengths you need. And there are also alternative methods such as size selection by precipitation, for which you can find the protocol in the documentation section of our website, along much more information on fragmentation and size selection. Now let's talk a little bit about sample QC, which is actually really important. Let's start with DNA samples. The first thing we want to do is to quantify the sample, and we recommend using qubit for this. We do not recommend using nanodrop for quantification because it might be affected by the presence of contaminants. We do recommend nanodrop to assess the purity of the samples though. So here on the top right, we have an example of the impact of phenol on library preparation. As you can see, the more phenol you have in your sample, the lower the library preparation efficiency will be. And we have assessed the impact of a variety of contaminants on both DNA and RNA. And this data is available on the documentation section of our website. For DNA, we also recommend you look at the fragment length, and this is because you'll need to decide whether you should fragment or size select your sample. In this example on the bottom right, we have one sample extracted with 7 different methods. And as you can see, for methods 1 and 7, the fragments are really, really long. So if you want to use a ligation-based library preparation approach, you would probably have to fragment those samples, sorry, 1 and 7, to make sure it is efficient. All of the other samples could be sequenced as they are, but if you wanted to deplete short fragments, then you could use size selection. So it's important to see how your sample looks so that you can make an informed decision on how to manipulate the sample to make sure you get the most out of it. For RNA, we also recommend using qubit and nanodrop for QC, and we recommend that you assess the RNA integrity number, or RIN, of all of your samples. RIN will measure how intact your RNA is, so it goes all the way up to 10. The closer to 10, the better. This is an example to show you what will happen if your sample is degraded. So we have used heat to artificially degrade an RNA sample, and this is why you shouldn't really heat your samples or subject them to harsh temperature variations. It's not good for them. So when we observe what we observe with what we've done of artificially degrading a sample is that lower the RIN, the shorter the reads. So you should always aim to sequence something with a high RIN because it's more intact, it has higher quality, and it will give you better results. You can sequence samples with a lower RIN than what we recommend, but keep in mind that that will result in shorter reads. Now let's take a look at the bacterial isolate sequencing as a worked example of the extraction step within an end-to-end workflow. Microbial isolate sequencing is a technique that provides important information for outbreak investigations, enabling characterization of antimicrobial resistance genes, virulence genes, and plasmids that can inform public health measures. It's also used in clinical microbiology research, food safety, and biopharma applications. However, if legacy short read sequencing technology is used for this, it is challenging to reconstruct complete high-quality microbial genomes. With a nanopore-only microbial isolate sequencing solution, or NOMIS for short, it's possible to generate complete reference-quality microbial genome sequences from a single streamlined workflow, making sure that no valuable information is missed. So let's look at the extraction step of the NOMIS workflow. As you can see from this flow chart, you can identify the extraction method you need depending on the experimental goals and the sample type. One option is to choose a universal bit beating based method of DNA extraction, which is here on the right, and this will enable extraction across different microbes of your sample. So it's a simple workflow, and this will give you read length N50 of 4 to 7 kb in sequencing. If, however, you would like to optimize for highly contiguous assembly of the genomes in your sample, you can go for an option that will give you higher N50s, as you can see on the left. And here we recommend different methods based on your microbial sample types. In this example, we're extracting from bacterial samples. So if we were going for highly contiguous assemblies, we would pick the bacterial genomic DNA extraction method, which is the NAB Monarch Spin Genomic DNA Extraction Kit. If the sample didn't lyse well, we would then be able to use the method for hard-to-lyse organisms. And here you can see the end-to-end workflow. So following extraction, libraries are then prepared using the rapid barcoding kit for preparation of up to 24 or 96 samples for sequencing in multiplex. For sequencing, a MinION flow cell is ideal, which is compatible with the MinION or GridION sequencing devices. Then lastly, the bacterial genomes data analysis workflow in EPI2Me enables de novo assembly, annotation, species ID, sequence typing, and analysis of AMR genes. The whole workflow is also fully automated on our Elysian device, which you'll hear more about in the next masterclass of this series. Next, let's take a look at direct RNA sequencing as a worked example. Capturing RNA diversity across gene expression, isoform identification, and RNA modification analysis can help elucidate molecular mechanisms, shedding light on applications such as disease research. With short read technology, though, the limited read length means that the transcripts generally can't be sequenced in full, limiting analysis to the gene expression level and missing important information on isoforms. It's also not possible to directly capture RNA modifications due to the need to convert to cDNA. In contrast, with Oxford Nanopore, a direct RNA sequencing workflow, you can sequence native RNA transcripts in single long reads, enabling characterization of complete isoforms and RNA modifications to reveal more information to elucidate those molecular mechanisms. So when it comes to preparing samples for direct RNA sequencing, if you're working with human cell lines, we recommend extracting total RNA using the Invitrogen Trisol RNA isolation reagent. Then to maximize output, we recommend using the Invitrogen Dinyl Dinabids mRNA purification kit to enrich for poly-A transcripts. And here's that step followed by the rest of the sample to answer workflow. For library prep, the native RNA is prepared for sequencing using the direct RNA sequencing kit, which doesn't require the PCR, so it preserves the native RNA. Next is high output sequencing of a whole human transcriptome on a Promethean flow cell, which is compatible with our Promethean 2 or Promethean 24 devices, depending on the scale you need. And then finally, the transcriptome's workflow in Epi2Me enables transcript identification, assembly, and annotation, as well as gene expression analysis and transcript use. Information on all the things I talked about during this presentation, so the extraction methods, size selection, fragmentation, and more, is available on the documentation section of our website. We have a lot of extraction protocols for both DNA and RNA, and a lot of sample types are covered. So please do go there and explore. Just to wrap up, hopefully after this masterclass, you will now know what to think about before you actually kick off your experiments. You need to know which type of data you need to generate, and that will allow you to decide how to extract your samples, how to make sure you preserve them in the best way. And hopefully now you know that even if your sample doesn't look as it should, there's further manipulation you can do, like size selection and fragmentation, to make sure you get the most out of your sample. Thank you for watching and happy sequencing.